Hi rabbits, Research Turtle here. Today, we're going to look at one of the most fascinating comeback stories in modern medicine. It's a story about a failed drug target that is currently rising from the ashes. We are talking about NHE-1 inhibition, moving from the ischemic paradox to the future of precision medicine. Let's start with the big picture. For decades, scientists believed that blocking the sodium-hydrogen exchanger, or NHE-1, was the holy grail for protecting the heart during a heart attack. But there was a massive crisis. In the early 2000s, a major trial called Expedition revealed a deadly paradox. The drugs saved the heart but damaged the brain, causing strokes. Development stopped cold. However, look at the right side of the screen. We are now in a renaissance. We aren't just blocking the target anymore. We're modulating it using SGLT2 inhibitors in cardiology, repurposing it for muscular dystrophy, and even using it to fight cancer. To understand why this matters, we have to look at the clinical gap. When a patient has a heart attack, we rush to restore blood flow. This is called reperfusion. But here's the irony. The return of oxygen actually triggers a second wave of injury called Ischemia-Reperfusion Injury, or IRI. This injury kills heart cells and leads to long-term heart failure. NHE-1 is the primary trigger of this damage. Let's zoom into the cell to see exactly how this happens. Think of this as a domino effect. Step 1 is ischemia. When the heart stops getting oxygen, the cell becomes acidic. This acidity activates NHE-1. The pump goes into overdrive, dumping acid out and pulling massive amounts of sodium, Na+, in. Step 2 is the switch. When we restore blood flow, the pH normalizes, but the cell is now full of sodium. To fix this, another pump, the sodium-calcium exchanger, NCX, runs in reverse. It kicks out sodium but pulls in calcium. Step 3 is the disaster. This calcium overload causes the mitochondria to fail, leading to cell necrosis. The takeaway? If we inhibit NHE-1 at the start, we stop the sodium influx, preventing the entire downstream cascade. Armed with this theory, pharma companies raced to find a drug. We went from the messy first-generation Amiloride to the highly selective second-generation Cariporide, and finally the ultra-potent Zoniporide. Preclinical models were overwhelmingly successful. It looked like we had cured ischemic injury. But then came the Expedition trial. This is the turning point. Look at the blue bars. Cariporide worked. It reduced myocardial infarction by 18%. The proof of concept was successful. Now look at the red bars. Cerebrovascular mortality, strokes, doubled. The drug saved the heart tissue but was toxic to the brain. The development was halted immediately. So what went wrong? Why the strokes? Our forensic analysis points to autoregulation. The brain is extremely sensitive to pH. NHE-1 helps regulate the pH in smooth muscle of brain blood vessels. When we completely blocked NHE-1 with high doses, we likely paralyzed the brain's ability to dilate blood vessels during low blood pressure. We also may have compromised the blood-brain barrier. There was also a dosing trap. The Expedition trial aimed for 100% blockade of a housekeeping gene. NHE-1 isn't just a villain, it regulates cell volume and pH for the whole body. By completely shutting it down, we disrupted systemic physiology. The lesson here? We need modulation, not elimination. This failure forced a strategic pivot. We're now moving away from blunt blockade toward 3 new strategies. 1. Indirect modulation. 2. Repurposing for safer indications. 3. Targeted nanomedicine. Let's look at the first success, SGLT2 inhibitors like empagliflozin. We know them as diabetes drugs, but they're powerful heart failure treatments. Why? Because they lower cytoplasmic sodium and calcium, effectively modulating NHE-1 without blocking it directly. They give us the cardiac benefit without the neurotoxicity seen in Expedition. Next, we have repurposing. Meet Rimeporide. Instead of acute heart attacks, it's being used for Duchenne Muscular Dystrophy, or DMD. In DMD, membrane instability leads to sodium and calcium overload, the same mechanism we saw in the heart. Rimeporide is showing great promise in reducing fibrosis, and because it's a chronic, lower-dose therapy, the safety profile is much better. Now, here's the most exciting new frontier: oncology. Cancer cells use the Warburg effect. They produce massive amounts of acid. To survive, they overactivate NHE-1 to pump that acid out. This creates a proton reversal. The inside of the cancer cell is alkaline, good for growth, while the outside environment becomes acidic. This acidic environment is a shield. It suppresses the immune system. By inhibiting NHE-1 in tumors, we can trap the acid inside the cancer cell, killing it, and neutralize the environment outside. This unlocks the immune system and makes drugs like PD-1 inhibitors work much better. Finally, how do we solve the heart versus brain toxicity problem once and for all? Nanomedicine. Scientists are developing smart nanoparticles that can carry NHE-1 inhibitors directly to the ischemic heart, bypassing the blood-brain barrier entirely. This could finally allow us to achieve the cardioprotection we saw in the early 2000s, but with zero stroke risk. So, to wrap up, the mechanism of NHE-1 remains valid. The failure of the past was due to complete blockade causing toxicity. The future lies in modulation, whether it's SGLT2 inhibitors for the heart, Rimeporide for rare diseases, or modifying the tumor microenvironment in cancer. NHE-1 has been resurrected. That's it for today. Turtle out.